So here's just a nice summary of how sound gets transmitted to the internal ear. Sound waves vibrate the tympanic membrane. The ossicles vibrate. Remember, the final ossicle, the stapes, will vibrate onto the oval window. So the fluid inside the cochlea is set in motion. It goes through the fluid. We didn't really name these things. The fluid is called perilymph. That's fine. If the frequency is not a sound that we can actually hear because it's above our hearing range or below our hearing range, well, it'll go right through those scalas that we really didn't learn the names of, and it won't disturb the cochlear duct. But sounds in our hearing range will go through that cochlear duct. Remember that little indentation of the cochlear duct? And then that will allow those hair cells to bend in your organ of Corti, which will perceive the sound or transmit that information to the brain to perceive the sound. So here's an illustration on how we perceive pitch. There's your cochlear duct inside your cochlea. Remember, the cochlea is actually spiral shaped, but they just made it straight so it's easier to look at. So here's a sound wave coming that disrupts the fluid. So you get this vibration in the fluid, and you see the cochlear duct interrupted. Now it can be interrupted at different locations. So it's saying that high frequency sounds will cause an action potential in the organ of Corti inside the cochlear duct near the oval window. But low frequency sounds will disrupt this cochlear duct near the apex with everything else in between. So this is how we hear higher pitch, middle pitch, and lower pitch sounds. Just think of like a piano keyboard, right, where you have low at one end keys all the way to high at the other end. That's our range of hearing from low frequency all the way to high frequency. So that's how we perceive the different pitches or frequencies of sound. So let's finish off the story. So there's our cochlear duct and our organ of Corti. So the impulse with those hair cells bend, since the impulse is sent to your central nervous system. And remember, everything except the sense of smell will synapse at the thalamus, but that's fine. The main point here, well, where is sound actually perceived? Well, we should know, yeah, the temporal lobe. Remember, the temporal lobe on either side is auditory and olfactory, and we're in auditory right now. So how do we process our sound? Well, remember, pitch is perceived based on the hair cells that are stimulated along the basilar membrane, but just think of that as the cochlear duct, and that's what we went through, the low pitch at one end, the high pitch at the other end. What we didn't go through yet is loudness is detected by the frequency or the number of action potentials. So a louder sound will send many action potentials, a softer sound just a few action potentials from that specific location. And localization of sound, how do we know where sound is coming from, is because we have two inner ears, two cochleas, one on each side, and it takes a slightly different amount of time for the sound to get to each ear, and that little difference of time, which is undetectable really if we're trying to hear a difference. It's too close together for us to perceive a difference, but our brain can, and that's how it knows which direction the sound is coming from. We're done with the cochlea, which contains the receptors for hearing, the organ of Corti. Now let's move to the middle portion of the ear, which is the vestibule. Now the vestibule has two little sacs in it. You see a little sac here, a little sac there. They're called the saccule and the utricles. That's fine. Those are the areas inside the vestibule. But so what's most important is what you see inside those two little sacs, the saccule and utricle. What you see in purple there, those are the receptors for equilibrium called maculae, or maculas one, but since there's two of them, put an E at the end, maculae. That's the receptors for static equilibrium. So basically, static equilibrium refers to gravity and changes in position of the head. So just think of like straight line motions. How do you feel yourself going up and down in an elevator? Well, these are the receptors for that straight line acceleration. maculae that are located in the vestibule. So static equilibrium is different than dynamic equilibrium, which we'll explain later. So static equilibrium, the receptors for it are in the vestibule. Where in the vestibule? In the saccule and utricle, which contains the receptors called the maculae. So again, the maculae are the receptors for static equilibrium. There's one in the saccule, one in the utricle. They monitor the position of your head, respond to linear acceleration forces up, down, or just horizontally accelerating. There's hair cells, just like there are in the organ of Corti, and the hair cells are bent as you accelerate linearly, and that's what produces the action potential. And there's otoliths, which are tiny little stones in the ear. Auto means ear, lith means stones, which is actually pieces of calcium which bend the hair cells. And we'll see that on the next slide, a picture of it at least. Here you see the maculae inside the saccule and the utricle right there. And if we enlarge them, well, there's your little hair cell. So when you accelerate up or down or horizontally, well, this membrane that has some weight because there's all these little stones or calcium stones on top of it, which are called otoliths, will bend these cells. And you can see that right here, which will produce the action potential. So here's an illustration inside the macula, or inside the vestibule, specifically in the saccule and utricle. We have maculae, which are these are the receptors for static equilibrium. And you can see right here, this person is just bending this head down. So what's happening? Well, these otoliths would have some weight. Force of gravity would pull them down, bending the little hair cells, which would send the action potential to the brain for processing that perception. And we have one final portion of the inner ear to go through, the semicircular canals. Remember, the cochlea is responsible for hearing, the vestibule for static equilibrium. Well, the semicircular canals are responsible for something called dynamic equilibrium, and that's equilibrium that has to do with, just to think of rotational movements, or you're spinning. You spin around and stop, you sort of still feel yourself spinning, right? And that's because of the receptors inside the semicircular canals. So at the end of each semicircle, these three semicircles, it bulges out. So that bulged out area is called an ampulla, and all the semicircular canals have an ampulla. Inside the ampulla are the receptors for dynamic equilibrium, and the receptors, well, each one's called a crista, or cristae with an E at the end for plural. Its full name is Crista Ampullaris because they're in the ampulla, but sometimes you just see them called crista. So here the head's rotating or spinning, which causes the crista inside the ampulla, those hair cells to bend. So same story as the macula inside the vestibule, but the crista inside the semicircular canals, those hair cells bends with dynamic equilibrium or spinning or rotational types of movements. And last point, I'm not too worried about all the stuff at the beginning here, but we should know something called motion sickness. It's when what you see, your visual input, doesn't match your input from your equilibrium areas, which is your vestibular apparatus. The vestibule for static equilibrium and the semicircular canals for dynamic equilibrium. Let's say, for example, you're on a large cruise ship. You're in a room, let's say, and you don't see yourself going up and down with the waves, although you are, albeit very slowly because it's a large vessel. So your visual input is saying, I'm stationary, but your vestibule is sensing the swaying of the ship. So your brain kind of is getting mismatched information from different parts of your body, which can cause you to get nauseous, or we call that motion sickness. Is your eyes are seeing one thing, but your vestibular apparatus, your equilibrium sensors, are telling the brain something else.